10 practice problems per chapter that you can have that represent what you're going to see in the exams. If you notice, 2 of those problems from the quiz, actually the chi-squared problem and the cross problem, were both from the back of the book, or both from the practice problems, with a couple of things changed within them. Speaking of that, exam 1 is not next week Monday, but the week after. My last office hours are on Monday and Tuesday next week, so start thinking about this early, please. Academic Mentoring Matters down here asked me to put all of their hours on Moodle. It looks like they have a lot of hours that they're actually around, so please use them. There's a spreadsheet on there for where you can meet them over there, and they may be coming in on Friday to talk for a minute or two, because I don't have very much experience with them yet. For Friday, I want you guys to read Chapter 5. What we're going to do today is kind of a little off of what we've been doing over the last couple days. This is going to be more of a research talk. I'm going to talk to you guys about what I do for my research. And the reason we're going to do that is because what we've been doing is talking about these Mendelian polymorphisms for the last few lectures. Now, very little of research is actually going out and crossing things into F1s and F2s, these monohybrid and dihybrid crosses. So I want you guys to kind of get a sense of what people do for research on these Mendelian polymorphisms and why they're particularly useful to use for instance, evolutionary genetics that I do. But we have a couple slides to finish up from last time. And when we finished up last time, we were talking about maternal effects. We were talking about the dextral and sinestral snails. But we were talking that maternal effects can be considered more broadly, where there's actually environmental impacts that impact the mother, that they can pass along information about that to their offspring. And I gave you this example in Campanula. This is a paper from Science in 2007. And what they did for this campanula study, Campanula is a genus of American bellflowers that you have out here in your forest as well too, is they grew the same accession of Campanula in sunny environments and in shade environments. They collected seeds and offspring from both of those, self individuals there, and they grew them both in a sunny environment afterwards. And what they found was if your mother was in that sunny environment, you ended up doing about 3 times as well than the accession that was grown in the shady environment, just because your mother was grown in a different environmental background. So why could that happen? There's a lot of reasons why that could happen, but 2 of them are described in your chapter. One is genomic imprinting, where you have differential expression of genes depending on inheritance from one parent or the other. And we'll talk about the molecular mechanism for that in 1 second. This is epigenetics, which I kind of hinted at when we were talking about history of genetics earlier. The second way you can do this is from cytoplasmic inheritance. So what we realize is we get 1 chromosome from mom, 1 chromosome from dad when we start off, but that's not true for mitochondria. Almost all of your mitochondria come from mom. Same thing with chloroplasts in plants. Almost all of your chloroplasts come from dad. I say almost all because we've just figured out in the last 6 months that there's a very, very small percentage of mitochondria that actually do come from dad. This is a science paper very recently. For the majority of your mitochondria come from mom. Okay, so how can genomic imprinting actually work? This is that epigenetics slide. You may remember this from the history of genetics lecture that we had. And epigenetics can happen through a couple of different mechanisms. First of all, we can have DNA methylation. In the human genome, this is really prominent on these CG regions, where we end up adding this methyl group from cytosine to 5-methylcytosine up on the side of here. The other way that you can have an epigenetic mechanism is through differential DNA packing around histones. So you can have some of these histones that create these heterochromatic regions where DNA polymerase cannot get into. Now, how do both of these things happen? When does methylation happen? When does histone packing actually happen here? It happens in response to an environmental cue in the parental generation. So the parent experiences a cue, typically, which releases a signal cascade and causes either methylation or histone modification. Note that in humans, it's mostly CG regions. Over 75% of methylations in CG regions in humans. That's not necessarily true in plant, fungi, or bacteria. They have slightly different patterns of methylation. So what happens between generations for each of these different environmental modifications? So normally, if we don't have any genomic imprinting, what we have is these are reset directly during the fertilization of the zygote, and again during development of that primordial germ cell in the mother. So the vast majority of these methylation marks, or these histone modifications, are completely reset in between generations. However, we've noticed recently, because of this Campanula study and others following it, that some of this we call transgenerational plasticity, the ability to respond to a cue that your mother experienced, occurs through these various mechanisms. Okay. I'm moving from that to the research portion of this talk. Does anybody have any questions on those first two slides right there? I know those are some fairly complicated concepts that are not found in your book necessarily.
What did you say about being reset?
Say again? So every generation, typically, these either histone modifications or methylation marks that we see, say your mother experiences a particular cue of interest, and you have this methylation mark that happens in response. During, actually, beyond meiosis and mitosis, when we're creating these gametes, those methylation marks are reset to cytosine in here. And there's a whole complicated molecular process that goes about doing that. But for a lot of species, that can be really beneficial, that reset, because if you don't experience the same environments as your mother, you may not want to actually start off with those advantages in life. Say, like your cubes or herbivorine plants. Yes, exactly. So that's one of the biggest places this has been observed in kind of the human model systems is in mice. And it's often the obesity mice are really interesting too, where they're fed diets either based on modern human diets or they're fed diets that are really stark in nature. And they have completely different epigenetic cues that happen after that. So,
if you wanted to study this and you needed, to amplify a whole bunch of DNA, how would you retain your methylation through PCR and through sequencing?
That's a great question. So where these methylation marks actually happen from, and we can talk about this more in the biotechnology section of this. How we do this is there's these bisulfide bonds that typically happen and keep these methylation profiles intact. So it depends on what restriction enzyme you use to cut up your particular DNA. So you can cut it up with a restriction enzyme that basically retains these methylation marks. And then after that, you can process it again with something that does not maintain those methylation marks. And you can see what the difference is, and that's how we determine what's methylated and what's not. All right, going into research, how do we use these Mendelian polymorphisms? So I'm going to talk about my research today. I'm going to give you a little context first in this. A lot of these terms will be things that we'll go over again in the beginning of the third section of this course when we start talking about evolution. But this kind of Mendelian polymorphism is going to run throughout this course. So what I study is the process of adaptation. That's how often does local adaptation actually occur in natural populations? And if it does occur, how does it occur? Why? What phenotypes, what genes, what environmental pressures are actually useful in it? The first question we know quite a bit about. We know that local adaptation is extremely common in natural populations. And we know that from are these experiments called reciprocal transplant experiments. And these experiments have been done since the 1940s, and they're really simple in nature. What we basically do is we go out to two differentiated sites. Let's say in this case a low elevation site and a high elevation site. We collect seeds from plants in those two different, from the organisms in those two different areas. And then we go out and we plant both the low and high elevation plants in a low elevation site and both the low and high elevation plants in the high elevation site there. And we compare how they do in terms of their fitness. And what we expect to see as a signature of local adaptation would be in the low elevation site, the low elevation plants are bigger and produce more offspring than the high elevation plants. In the high elevation site, we'd expect that the high elevation plants would be bigger and produce more offspring than the low elevation plants. Now we've done this kind of study with many different kinds of organisms, not just plants, over 1,000 times, and meta-analysis suggests that adaptation happens over 70% of the time in these particular studies, which means it's really frequent in natural populations. What we know less about, actually, though, is the mechanisms that actually underlie local adaptation in those populations. This is a guy named Ronald Fisher. He's kind of the architect of our field, and he came up with a lot of these questions during the modern synthesis in the 1920s and 1930s. And what he wondered about, and what I wonder about in my research, is are there generalities in this process of adaptation? Are there generalities in the targets of selection that occur between these different evolutionary adaptation events? That is, are there differences in the number, effect size, or type of genes, mutations, and phenotypes that occur in adaptation? Second, he asked, Are there generalities in the agents of selection that are actually acting among these disparate groups of organisms? That is, in the type of selection pressure, the duration that selection actually occurs, and the strength of selection, so how strong selection is for any given generation. And all of these are getting around really understanding the predictability of evolution in general in these natural populations. How quickly can adaptation occur? How convergent is this? How predictable is that process? And how constrained is that process? We'll talk about that too. The reason these questions are really difficult to answer is something that I've kind of gone at in this course thus far a little bit. So to understand adaptation, we have to understand how variation in genotype translates to variation in phenotype, what we've been talking about so far, and how variation in phenotype in a given environment translates to variation in fitness. So the kinds of information we need for each one of these different transitions is very different. For the genotype to phenotype transition, we need a model organism that is really well known genetically, that we know a lot about the genome sequence, about the different genes involved in it. For this phenotype to fitness transition here, we need a model organism for ecology, something we can plan out in the field and actually observe how phenotypes translate into fitness in those different environments. Of course, it's not actually as simple as one environment here. When I was telling you about the reciprocal transplant experiments, we were talking about looking at how two different populations actually occur over a gradient. So really we're thinking about these different transitions in multiple different environments. One of the ways we can simplify this a little bit is by using environmental gradients to study this adaptation, such as this mountainside right here. Now if you can imagine a single plant that exists across this entire elevation gradient here along Mount Baker in western Washington. Now along that particular gradient, you'll notice there's a gradient in a bunch of different environmental conditions. Temperature, precipitation that falls as snow, seasonality, a lot of things are going to differ across this gradient right here. And you can imagine that single species that's distributed across all those different environments, there's going to be a phenotypic gradient as well too, for many of the different traits of that species. Here I'm just representing that by plant height necessarily there. And if we compare those two things together, we can get what we call a cline, which is a shift in genotype or phenotype over an ecological gradient. Here we have plant height and temperature on the x-axis. And what those clines actually represent is a prediction about how natural selection is acting in those populations. So for instance here, we might predict that taller plants have higher fitness in warmer environments and smaller plants have higher fitness in colder environments along this particular gradient. Now it's not a very good prediction because there are a lot of different environmental variables that vary along those environmental gradients. I just told you temperature, precipitation, seasonality, all of those different things. But it might be a slightly better experiment if instead of doing this just once, if we could look in several different geographically disparate areas spread across the world where that same plant exists and compare those different gradients as replicates of the evolutionary process. And that's what I'm going to talk to you about today. How we can use kind of these replicate ecological gradients in the context of this phenotype called cyanogenesis in white clover to answer some of those major questions about adaptation, about the targets of selection, the agents of selection, and the predictability of evolution in those populations. Now this phenotype called cyanogenesis in white clover I mentioned once in this class already. Cyanogenesis is the ability to produce hydrogen cyanide after tissue damage. Over 3,000 different plant species do this, including many of the crop species that we think about. Sorghum, cassava, these things that are major crop species, they actually have to have the hydrogen cyanide leached out of their grains or tubers before you can actually eat these plants. Peach pits, apple seeds, many of these different things produce hydrogen cyanide. We study this in white clover because instead of having a quantitative variation in cyanogenesis, that is, all plants within that species produce cyanide, they just produce it in different levels. White clover has this in a defensive polymorphism. That's Mendelian polymorphism, such as that we've been talking about. So out of 3,000 plant species, we know of 2 that have cyanogenesis as this Mendelian polymorphism. Now, how do we get that polymorphism in white clover? So this presence-absence polymorphism of cyanogenesis occurs based on the presence-absence of 2 biochemical components. You have cyanogenic glucosides, which are basically here a sugar molecule, a glucose molecule, with a cyanide group attached to it. And we have this enzyme called linamorase, which is an enzyme that's capable of breaking apart these cyanogenic glucosides and essentially cleaving off that cyanide group, so we produce hydrogen cyanide. Now, hydrogen cyanide, as many of you probably know, it reacts in the mitochondria and basically eliminates cellular respiration for most animal taxa, and kills you very quickly, surprisingly. Okay, so we know something about the inheritance of these two different biochemical components too. Each one of these is encoded by a single Mendelian polymorphism as well. So the presence of cyanogenic glucosides is encoded by this AC locus, where the dominant AC gene encodes the presence of cyanogenic glucosides. Likewise for linamrase, if you have one big Li allele, you're able to produce linamrase. So the dominant allele in this linamrase case is big Li. Now, what does this mean in terms of the cyanogenesis phenotype? Let's run through this because the entire talk kind of depends on this. If you have one copy of the big Li allele, and you have one copy of the big AC allele, you're capable of producing hydrogen cyanide. Just like those examples we were talking about with epistasis earlier, if you don't have one big AC allele, if you don't have one big Li allele, or you don't have either of these in a plant, you're not capable of producing hydrogen cyanide. So there's 3 different genotypes that you can't produce hydrogen cyanide, you're not cyanogenic, and one where you can. Now, from our last talk, this is the example of double recessive epistasis right here, where we would have a 9:7 ratio in our F2 generation if we were to do this. The nice part about the cyanogenesis phenotype, though, is we can survey it really easily with something we call Fenger-Enger assay. Now, basically, this uses a colorimetric paper that changes into a blue color whenever there's hydrogen cyanide present. And you can picture we have a cell well under here, where there's a bunch of different wells. What we can do is basically just put tissue from those plant in those wells. And if we add just tissue to those wells and we mash it up to simulate herbivory, we might get blue if that plant is cyanogenic in nature. We can also look for the presence or absence of cyanogenic glucosides and linamrase too by adding exogenous compounds. So if we add cyanogenic glucosides, which is what this linamrase is, then we can test for the presence of that enzyme for linamrase. If you have this here, after we've added cyanogenic glucosides, this means you have the enzyme, but no cyanogenic glucosides that you're able to produce. So you have at least one big Li allele. Now the other way you can do this is, again, if you add linamrase, you can survey for the presence of cyanogenic glucosides or not. So if you had a blue allele here and no blue coloring in either of the other wells, you would say that we have cyanogenic glucosides, but you're not capable of producing the enzyme here. Now this is what we call an adaptive polymorphism, because it has alternative states that encode higher fitness in different environments. How do we know that? Oh, I'm sorry, we're not moving quite to that yet. We can talk about this at the Mendelian level, but we can also go very much deeper into this too and talk about how these Mendelian polymorphisms actually occur at a molecular level. Okay, so, sorry, we got a little bit, my animations are getting ahead of myself here. So for the AC gene, this dominant AC allele is basically the gene CYP79D15. The gene name doesn't actually matter. What matters is that this particular gene encodes the enzyme that's the first step in synthesizing those cyanogenic glucosides. For moving from amino acid, phenylalanine, to these cyanogenic glucosides, you have to have this gene right here. To get the recessive allele here, where we're not able to produce cyanogenic glucosides, we have these gene deletion events. So actually, this is not one gene, but it's 2 genes in tandem right next to each other. This depends on copy number variation. And we have a gene deletion polymorphism that spans this. So the recessive alleles do not have a copy of this gene in general. Basically, the exact same thing happens at this Li locus. So the Li gene is encoded by a single protein, which is the Li locus. It also has a gene deletion polymorphism at it. So if you have one copy of that dominant Li locus, you have those 2 genes. If you do not, if you have that recessive allele, that means you do not have this gene at all. It's completely cut out of your genome. And if we look at this, it's actually a bit more complicated for AC than cutting out one gene. That gene deletion spans a large portion of the genome. How many of you guys have seen a diagram like this before? Anybody? A couple of you. Okay. So this is what you get out of a genome browser. If you're looking at the human genome, you can go online and you can pull this up. What this represents is a segment of genome from here that goes from 0 kb to 250,000 bases here, essentially. And you have these little regions in here encoded by little arrows that are individual genes here. There's exons in the middle of those genes that we don't have in there as well either, that you can typically see if you go down to a finer scale on this. So that AC gene deletion, we know actually doesn't just delete that CYP79D15. It extends much farther and actually deletes 3 other genes that we know of too in that system. So I've been saying that this is adaptive. How do we know that this is adaptive? The first evidence that we had was actually from a population geneticist named Hüner de Day in the 1950s. And what Hüner de Day did is he had a lot of friends that were spread out all over Europe. This is where white clover is actually native to, the Eurasia region. And he asked all of those friends to send them 15 or 20 white clover samples from their area. And he did that Fengel-Egner assay on all of those samples. And what he basically found was this, that in warmer regions, say in Turkey or in Spain, getting up here in France, almost all the plants that he surveyed were cyanogenic. But if you start getting into colder areas in Eastern Europe, up here into Russia and in Scandinavia, he found that none of the plants were really cyanogenic here. And what this produces is a relatively nice climb across Europe here from warm to cold regions, where you have more cyanogenic plants found in warmer regions. This is one where I came into the system. I came in and I was looking for a product to do it for my PhD. And my first thought was, well, white clover was introduced to the United States less than 300 years ago. I wonder if a similar cline has evolved across the United States in that 300 years. This was relatively controversial at the time because evolution in 300 years is a relatively rapid evolutionary event. So I went and surveyed populations from Louisiana all the way up to Wisconsin up here and phenotyped them for cyanogenesis. And what we found was a very similar pattern to what was in the native region in Europe, where in Wisconsin, basically about 14% of the plants produced hydrogen cyanide, and in Louisiana, about 85% of the plants actually produce hydrogen cyanide. So it's a huge difference and a nice cline across there. But when we were thinking about this, we were thinking about, hey, this cline could actually be produced by 2 different ways. One of the ways could be just through natural selection and gene flow, like I've been talking about already. That seems like the simple explanation. But there could be another even simpler explanation, really. If you were moving to Wisconsin and you were coming from a cold region, and you knew you were going somewhere cold, you'd probably take the white clover plants that were already adapted to the cold area where you're at and bring them to Wisconsin. Likewise for Louisiana, if you're coming from somewhere that's relatively warm, and knew you were going somewhere warm, you'd take the plant that was adapted to those warm areas over here. And that, you could just be bringing plants from either cold or warm regions in Europe straight to the United States, and you could be recreating this cline without any natural selection happening whatsoever, just simply through gene flow coming from Europe over to the United States. How can you test for that? There's a really easy way to test for that, actually. And we're doing that using these neutral markers called microsatellites, where these base pair repeats that mutate relatively rapidly. So we can look at how long these fragments are of these base pair and use those as different alleles here. We can look at this graph. So on the bottom axis, we have divergence of minimum winter temperature, looking at each one of these populations across here. So you imagine that things are closer together, have less divergent minimum winter temperatures. On the y-axis, we can look at genetic divergence. So say the genetic divergence at that cyanogenesis, those 2 cyanogenesis loci, is going to be less at populations that are close to each other, and is going to be greater divergence at populations that are further away, because we see that cline occurring across that latitudinal gradient. Now, if we were to look at those microsatellite markers and see a very similar pattern to what we saw at that cyanogenesis, those cyanogenesis genes, we'd probably say that's an artifact of introduction, because all that happened is everything that's occurring in the genome has that same clinal pattern that we observed at our cyanogenesis genes. However, if those neutral markers say something different, if they're not under unnatural selection whatsoever, and the cyanogenesis genes are, we might see slope of that line being much less over here, and there being a much smaller clinal pattern across that entire gradient right there. And that difference between the neutral markers and these functional genes would suggest that natural selection is actually acting in these populations. So this is what we actually find when we do this, these different lines. So the blue line down here is our microsatellite markers. And each of these three genes here, the red is the ability to produce cyanogenic glucosides, the green is the ability to produce linamarase, and the black is the ability to produce hydrogen cyanide. And what you can see is the slopes are very different between those lines. And that suggests that natural selection actually has occurred in the United States in those 250 years since introduction. And that's what you guys should have been reading about in that paper, that I assigned for today. Okay. But there's other patterns of selection that could be occurring here too. We understand that there is similar patterns acting across these huge latitudinal gradients that are creating these clines in each one of those areas. But there could also be some patterns that act within populations. One of those patterns is what we call epistatic selection right here. So it's taking those ideas we learned about epistasis and Mendelian genetics and applying them in selection. So we have to think about this from an ecological concept. Which particular genotypes are going to be beneficial in these environments? So if cyanogenesis is really selected for, we think cyanogenesis would probably be selected for here. And we'll represent that by plus signs. But if we don't think that the cyanogenic glucosides or the enzyme have any other purpose besides for creating cyanogenesis, if you produce either just cyanogenic glucosides or linamarase alone, that would suggest it's wasted effort right there. That you're putting all these things into metabolism that really you could put into creating more offspring or something else. So this might not be beneficial for you to do. So we could think that the two genotypes, looking at one of these epistasis squares I was showing you earlier, not a Punnett square, that the highest fitness would be had by plants that produce both of those compounds or neither of those compounds. And this might be a pattern that we expect to see within those populations. This is what we call epistatic selection. We'd have more of these genotypes in those populations than we would expect just from the actual allele frequencies in those populations, and less of these other two genotypes here. The opposite of that is if both cyanogenic glucosides and linamarase had some beneficial function, but cyanogenesis actually had no beneficial function, and there was some selection over here too. We can grade this on a scale of LD, so linkage disequilibrium over here. This pattern over here represents an LD of 1, and we'll go over the math for LD in about a month. This represents an LD of -1. So if we'd expect to see epistatic selection within these populations, we would expect to see LDs of 1 here. When you look across all the populations that today surveyed in many different regions, he finds this pattern of epistatic selection extremely often in those populations. All those red arrows, I know it's hard to see those small numbers that are coming up here. But there are some populations where he's found, in 2/3 of the populations he surveyed, he found some version of epistatic selection. Does that same thing occur in the United States? And the answer is no. Our linkage disequilibrium is almost exactly at 0, which means we don't see that pattern where those two particular cyanotypes are actually selected in those populations. And that means there are different patterns of selection between these two different transects. Evolution is acting in different ways in these different places, which is kind of an interesting thing as well. So now I've talked about the targets of selection. The difference in cyanogenesis is created by these two different Mendelian polymorphisms that occur because of gene deletion events at the molecular level. Let's move on to agents of selection and think about what selection pressures actually drive adaptation in this system. This is one of the reasons why these Mendelian polymorphisms are great to study. We can actually do experimental manipulations with these. So, backing up, cyanogenesis, let's talk about the ecological selection pressures that actually create these variation in these cyanogenesis clines that I've been talking about. So the cyanogenesis, as I kind of alluded to earlier, has a cost as well as a benefit. You're putting energy into producing cyanogenic glucosides and linamarase. Thus, if you're in these really cold areas where there are few herbivores and no growing season, it might not be advantageous for you to produce a defense against herbivores. There might be no reason that might have any effect on fitness. And therefore, acyanogenic plants could be selected for in those cold areas. But if you're in those warmer areas, where there's normally more herbivores and there's longer growing seasons, cyanogenic plants might be extremely beneficial there to have that herbivore defense, and it might not be a waste to put energy into those two different compounds. This is a long-held view about why these cyanogenesis clines actually exist. It's been over 30 different studies looking at cyanogenesis and herbivory in the field and looking at these resource allocation trade-offs. We know these two things exist, and we know that this could be a potential cause for these particular cyanogenesis declines. So one thing that won't surprise you, I really like to travel as well. That's one of the reasons I went into this job, because I got to survey cyanogenesis declines not only in the United States, but also occur in New Zealand and in the North Cascades up in Washington. And that's where I spent the majority of my time. And the reason we chose these places to go look for other cyanogenesis declines was that because these areas have mountain ranges that cross the same difference in minimum winter temperature that we see occurring across the central United States, except they occur over much different size gradients. This one about 100 kilometers in New Zealand in the Southern Alps, and this one over about 10 kilometers on Mount Baker that I showed you earlier. Now the question is, do we see similar cyanogenesis declines in these different areas? If you remember, this was the pattern I showed you between neutral markers and the cyanogenesis genes earlier. So what do you see in New Zealand and in the North Cascades? Well, we see different patterns in each of these transects. In New Zealand, we see there's a significant decline in the ability to produce linamrase, so that big Li allele. And we also see a decline in cyanogenesis here, although weaker, still greater than the neutral marker background. In the North Cascades transect, we see there's a decline in cyanogenic glucosides and a decline in cyanogenesis, but no decline necessarily in linamrase like we saw in New Zealand. So why is there these difference between these different transects? One of the explanations for that could be that the only thing that's actually selected in any one of these different transects is the ability to produce hydrogen cyanide. And it doesn't matter if you produce cyanogenic glucosides or linamrase, as long as you can get to the point where you're producing hydrogen cyanide. And thus we might see those different patterns and those biochemical precursors between those. But there's a second explanation too, and that's that each one of these two cyanogenic glucosides or linamrase could have alternative functions as well too here. So they could act as different phenotypes besides just for producing cyanogenic glucosides. This is what we call pleiotropy. And we have a good idea that it's that second thing actually, that these cyanogenic glucosides have an alternative function. About the same time that I was doing this work on hydrogen cyanide, a different group of researchers was doing work on sorghum and cyanogenesis. It produces a cyanogenic glucoside called durum. And durum can act through this particular wounding mechanism also to produce hydrogen cyanide as a defense against herbivores. But in this biochemical network that we see here, durum can also have a different function where it can be turned over through a series of enzymatic interactions into ammonia. You won't know this about ammonia because most of you probably haven't studied plants at all, but this is the primary source of nitrogen for plants. Plants use ammonia for all their functions, so it's the main thing that they convert into other sources of nitrogen. The other thing that you may not know from this is the first thing that happens during drought stress is not actually a water limitation, but they cut off the enzyme that transfers other nitrogen products into ammonia. This is not something I'm ever going to ask you at any point because you guys probably will not be plant scientists, but notice that this could be beneficial. This ability to produce ammonia from cyanogenic glucosides could be beneficial if your plant is stressed during drought, and it may be a different reason that you'd want to produce cyanogenic glucosides. And then we look like our patterns across these declines kind of correspond to this fairly well. If you look across New Zealand, there's a lot of variation in aridity index, indicating there's probably a lot of difference in drought stress. And there's also a difference in the frequency of producing those cyanogenic glucosides, where places in drier areas produce more cyanogenic glucosides than plants in the mesocaric areas. We don't see that same pattern in the central US or in the North Cascades because there's almost no variation in aridity in those two different areas. If you look from Louisiana to Wisconsin, we all get a lot of precipitation across that same gradient, although at a little bit different times of year, actually, too. But we don't see that same pattern in between cyanogenic glucosides and aridity index there. So what do we do when we think we see a pattern? We go out and we test it with an experimental manipulation. In this case, we're doing a common garden experiment. We're bringing all of these different genotypes into a single garden and looking at how they do in terms of fitness in the absence of herbivory. So we took a bunch of seeds from what we call F2 SIB crosses. The point of doing these different crosses and creating an F2 generation here is that we can segregate the cyanogenic glucosides and the linamrase while we manipulate the genetic background. So we basically are trying to get rid of background effects from any other loci throughout the genome by doing a couple of crosses and growing a lot of plants in these environments. And we can put these in two different treatments, one being a water treatment, because that's what we're interested in testing, and one being a fertilizer treatment, because that impacts the ability to uptake nitrogen. What do we see in these experiments? In the standard water treatment, if you look at these different phenotypes that we have up here, there's no difference between those different phenotypes in their total reproductive investment and how many offspring they actually can produce. There's huge error bars on this. Anytime you see error bars that overlap with means of other things, that means that there's probably no statistical association between them, as a rule of thumb. However, in the low water treatment, we see that plants that produce cyanogenic glucosides because they're cyanogenic or because they're big AC little li plants have higher fitness and higher reproductive value than plants that do not produce that. So you're looking across here and you're seeing that these box plots are fairly similar. But the mean in these two different box plots is over 200 more seeds are produced in the low water treatments for plants that produce cyanogenic glucosides, which is a huge difference. That's over a 30% fitness advantage. That's a similar fitness advantage to what we see for when we actually expose mice to warfarin, what we use to kill them, essentially, which is really interesting. Okay, so from this common garden experiment, we can make some new hypotheses about why those cyanogenesis clans actually evolved. That it could be strictly from abiotic conditions associated with water and fertilizer, rather than these defense against herbivores. And in this, defense against herbivores could be a primary or a secondary function that also occurs, but there seems to be multiple selection pressures working here. To confirm this, we're going to bring this back out into nature again. We've been looking over gradients and minimum winter temperature. What if we hold minimum winter temperature constant and look across just a gradient and aridity? And as you probably know, as you go from Louisiana over through Texas, you have a very large aridity gradient there. And that's actually where we looked. We collected plants from different locations, from Memphis over to just west of Tucson. And what we found was this. In those drier areas, plants, populations that were in drier areas produced more cyanogenic glucosides, exactly as we expected, but had no difference in cyanogenesis or in the ability to produce linamers, suggesting this is a major selection factor. So what did I just tell you? I told you that there are multiple targets of selection in this system. We had the ability to produce cyanogenic glucosides, the ability to produce linamers, and the ability to produce hydrogen cyanide in the system as possible different targets of selection that could be acted upon as phenotypes in those populations. I told you that there are multiple different agents of selection that could be acting upon those that create differences in fitness in different environments. You see, herbivory could possibly act to select for plants that are cyanogenic, and actually cyanogenic glucosides are herbivore deterrent too, which is something I hadn't told you before. And we think drought is actually acting to select for cyanogenic glucosides and the production of hydrogen cyanide as well. There could be interactions between this. So what this is saying is this is a much more complicated system where there are multiple agents and targets of selection that underlie the process of adaptation than what was originally suggested, where we just had this nice defense against herbivores that was a single Mendelian trait. What this is telling you is that we have these Mendelian factors, but they add together to create multiple phenotypes, and they have a lot of different multitude of selection pressures in nature that can be used to create really interesting recurrent patterns across the globe. So in each of these different areas that I've highlighted with those boxes right there, we see cyanogenesis clans evolving from with more cyanogenic plants in warmer areas. So what we see after introduction is these recurrent patterns arise despite the complexity in the process of evolution. Now I'm going to go for just a couple of minutes into the predictability of adaptation and thinking about the evolutionary origins of the cyanogenesis polymorphism. And then I'm going to give you a couple of minutes to ask me questions about this system too. And no question is a bad question here because I understand this might be a bit overhead sometimes. So one thing I didn't tell you, I told you that cyanogenesis arises in over 3,000 different plant species. 3,000 species are cyanogenic. If we look just within this trifolium genus, there's actually 200 plants in the trifolium genus. And in the European clade of that, we can survey those plants to see if they're cyanogenic too. And what we find is we can get a tree diagram like this coming out of it that shows the evolutionary history of these different species. So each of the different species in this trifolium genus, in one section of it at least, are represented here. And I've put down if they're able to produce cytogenic glucosides and if they're able to produce linamarase for each one of these species. Now what you can see across this tree is that there are multiple places where species can only produce cytogenic glucosides, one species where it can only produce linamarase, another species, Trifolium ismulcarpum, which is not Trifolium repens, which is the white bug we were talking about, which is also cyanogenic. And then this one down here, this allotetraploid, Trifolium repens, is the one we've been talking about so far for cyanogenesis. The question is, what does this tell us about the evolutionary origins of cyanogenesis? One of the things it tells us is because these are two different clades here, that the ability to produce hydrogen cyanide has arisen multiple different times at different points in evolutionary history. And we can see that because these aren't sister species to one another where it just arose once and had this pattern of evolution occurring here. Even within white clover, we can see that cyanogenesis has arisen more than one time. And we can look at this we can tell this by looking at the flanking regions of those two gene deletions where we saw for cyanogenic glucosides and linamarase. So if you look at these recessive plants that have these particular gene deletions, if cyanogenesis, if this gene deletion only occurred once, what we would expect to happen is we expect to see one single size of deletion here. We'd expect to see the same flanking sequence in each one of those particular plants that had this gene deletion. And that's not actually what we see. If we amplify a bunch of regions in the flanking regions of these genomes, what we can see is there's multiple different flanking region sizes here. Some that differ by upwards of about 300 different base pair there. And this suggests to us that the cyanogenesis, the deletion at this particular gene has actually occurred not once, but multiple times. The same thing we can see for linamarase if you look at the flanking regions of the genes that produce linamarase, that have these gene deletions. It's a little bit more complicated in linamarase. We see a second deletion that actually occurs downstream of linamarase. And we see multiple different sizes of the flanking regions there too when we amplify across this particular gene. This suggests the same thing, that this linamarase gene has been deleted multiple times, and that this Mendelian polymorphism has been created multiple times in history. Okay. So why? Why would we see all of these different gene deletions occurring at this one point? Why would they happen multiple times like that? That's not something that we expect to see in evolution. We expect to see evolution happening over a long, gradual process, but maybe not so much anymore. One of the reasons that we see these deletion events occurring is because in these flanking regions where you see these different gene deletion sizes, there are all kinds of different repeats of between 150 and 200 base pair repeats in there that occur over and over again. And one thing we know from these repetitive regions is there is a greater frequency of this poor coupling during meiosis and mitosis here. So what happens here is potentially this blue region could accidentally pair with this green region. They are the same sequence there, relatively. So you might get a hairpin loop that occurs that ends up being excised from this DNA pattern through mechanisms to prevent these kind of mistakes from occurring during meiosis and mitosis. And by doing that, you get these gene deletion regions that occur in neighboring regions. And this is something that we found that is incredibly prevalent. And this is something that's only occurred in the last couple hundred years, or I'm sorry, a couple decades, that we started figuring out where these repetitive regions lie and seeing that there's a lot of polymorphism in gene deletions and insertions right next to these kind of regions. Okay, so what I tell you today before we take some questions here. I told you that these cyanogenesis clines evolved rapidly after introduction, but they occur through slightly different evolutionary mechanisms in each one of those different places in New Zealand and the North Cascades. I told you there are multiple different selection pressures that are used for creating these clines. Not only herbivory and resource allocation, but also talking about drought stress that we're looking at there. And finally, I told you that there are these recurrent patterns, these cyanogenesis clines that evolve across the world, despite the complexity in the selection patterns that you have, and have arisen multiple times in evolutionary history, largely because of these repetitive regions that occur right flanking these particular genes of interest that correlate together into producing these cyanogenesis phenotypes. So, with that, I'd like to ask you guys about what questions you have in this system and see where we can talk through some of these things.
For those simple sequence repeats and your microsatellites and stuff like that, could you explain that better? Because
Yeah.
I'm unclear as to how you use that to actually tell whether it was a byproduct of like bringing it from Europe or if it was like genuine evolution.
Okay. So when we look at what we call neutral markers throughout the genome, these are things that should be evolving without constraint whatsoever. These microsatellite markers that I'm talking about are repeats of 3 particular base pair. They can be any 3 base pair. But these tend to occur throughout the genome, and over 25% of our genome is encompassed by these small microsatellite or mini satellite repeats. Now the nice part about them is because they have all this repetition in those regions, and this exact same mechanism that I'm talking about here, they tend to mutate about every 100 generations. You see slippage and you get another 3 base pair repeat, or you cut out a repeat there. So we can use this to genotype different populations and see how related these are to one another. This is the same technology we use for DNA fingerprinting when we're trying to assess crime, essentially, and trying to identify an individual. If you have enough of these 3 base pair repeats, these microsatellite loci, distributed across the genome, everyone will have a different individual signature at those 3 base pair repeats. And we can look at different populations and see, hey, in this population, we have almost all the individuals have 10 3 base pair repeats or 11 or 12. In Wisconsin, per se, we can say that almost all those individuals have 15, 16, or 17 3 base pair repeats. And we can infer that there is some distance between those 2 populations because of those differences in those neutral markers that just occur because those are independent populations that are breeding together, that are breeding independently in those 2 different areas with limited amounts of gene flow connecting them. Alternatively, in the white clover system, where there's a ton of gene flow because everyone drives cars, wears soccer cleats, and runs around the country fairly constantly, there's a lot of gene flow in the system. So what you see is if you have 10 to 12 repeats in Louisiana, you see also in Wisconsin, there is mostly plants with 10 to 12 repeats there too. So there's very little signature of what we call population structure across that entire gradient. However, what you do see for the cyanogenesis genes is you see that strong cline occurring, where there's like 10% cyanogenic plants in Louisiana and like 85 in Wisconsin. So we can directly compare those 2 things because the cyanogenesis phenotypes are just those 2 genes as well. And by seeing that difference there between the neutral markers and that and the actual functional phenotype, that suggests that there's selection occurring across that region. Does that make sense? Anybody else?
Yeah.
So you said cyanogenesis could be like wasteful in colder environments. Could it be also like harmful because it would like kill the herbivores that are like distributing, like for fruit, like apples that have like hydrogen cyanide? Would that like kind of harm their fitness in a way?
In apples, one of the reasons that cyanogenesis evolves actually in the seeds is because they want certain things to eat those seeds but not other things. So the amount of hydrogen cyanide that's produced in the seeds of apples is not necessarily enough to deter mammalian herbivores, but it is enough to deter small insects that can be eating them. So, but similar concepts could occur too from pollinators as well. If you produce cyanide in your leaves, maybe you also produce it in your nectar. Any other questions? I'll take one last one. Yeah.
So the variation, I think you said a variation is already explained.